Welcome everybody to this month's Genomics Lite titled Genetic Alchemy, and we've got Nneka with us, the head of gene editing at the Wellcome Sanger Institute, to talk with you this afternoon. And my name is Sam. I'm a science engagement officer at the Wellcome Genome Campus, and I'll go through a little bit about where we are in the country in just a second. So for those that are brand new to Genomics Lite, our events aim to help everybody to explore the science of genomics and what it means for our lives. We're going to have some bits where people are, participating in polls. We're going to have a Q&A. So we just ask that everybody participate in the spirit of curiosity and respect any different views, identities, or experiences that might come out in any of the questions that might get asked, or any comments or anything like that. I will be moderating the platform this afternoon so that it's safe and inclusive for everyone. If you've never used a webinar before, this is just quickly how it works. So we've got a chat button. Use this if you're having any technical issues this afternoon. I will do my best to be able to fix those. I can't promise that Zoom will necessarily behave with every technical issue that might occur, but I will do my utmost to help. So if you're not hearing anything, you can't quite see the slides or anything like that, if you put any sort of technical issues in that chat, or any questions just generally about something, put them in that section and I will get to those. There's a button that's called closed captions. So if anyone would like subtitles with this afternoon's talk, if you just click that button, it will give you a transcript just below the slides to go alongside the talk. And then, as I say, we've got a Q&A session that will happen just after the talk from Nneka. So please do put any questions that you have, or anything that comes to mind that you might want to ask. Just put them in there the moment that they come to mind, and we'll get to them when we get to that section. Now, this is where we're based. So this is our campus. We're based at the Wellcome Genome Campus in South Cambridgeshire, just outside a little village called Hinxton. This is taken on a really nice sunny day that we are not blessed with having today. But yeah, on a nice day, that's what it looks like. So yeah, this is where we're based at the Wellcome Genome Campus. And as I say, I'm a science engagement officer. So my role is to basically try and help share what we do on our site. And I'm really, really grateful to be joined by Nneka to help me do that today. So this is a little bit about how it will work. So we'll have a talk, then we'll have a Q&A, and it will be wrapped up by 5:30. The session is being recorded and it will be available on our YouTube and our website very shortly after the session. So if for any reason you need to drop off, or your internet or connection drops off at any point, this will be recorded, so you will be able to catch up at any point. So yeah, without further ado, I'll hand over to Nneka. So I will stop sharing and pass over.
Thanks, Sam. I will start sharing.
Perfect. Yeah, that's all come through.
Fantastic. Okay, so hi everyone. Thanks for joining us today. Like Sam said, I'm Nneka, head of gene editing, and today I'm going to talk to you about transforming the genome. So let's get started. So first, I kind of wanted to explain the title a little bit. So when I chat to people about what I do, often like the response is as if I do some magic. And so kind of I've always thought about transforming the genome as alchemy, kind of that transforming one thing to another. And kind of when having this title, I kind of wanted to explore it from that angle. So today, what I'm going to talk to you about is first start off a little bit about my background and my role at Sanger, then some basics on the background behind this alchemy. Then I'm going to talk about the work we do specifically at the Sanger. And then in the latter half, I'll start to explore this beyond the work we do in Sanger, look at the therapeutic potential of transforming genomes, what you might see beyond the lab, outside of clinical work, the type of things being used, the ways they've been used to do things outside of a clinical setting, sorry. And finally, touch on machine learning and how that's actually becoming more and more a really important part of this field. So quick stop about me. That's me looking very sciencey. So this is my team. I head the gene editing team. And again, it's based in this lovely Green Institute near Cambridge. And this is my team. I'm hoping you could see the pointer, but if not, I could change it on a more gloomy day, but it's my fabulous team. And so wanted to kind of chat to you a little bit about what we do, because we're slightly different from a lot of the people or the scientists who work here, because we don't work on primary research, rather we're a core facility. And what that means is different researchers across the campus or across the institute will come to us with a biological question. So perhaps they want to explore a particular gene where mutation is known to contribute to a brain tumor, for instance. And so they might need us to convert a healthy cell into a cell that has that tumor and then has the mutation, sorry. And then they could do the downstream elements on how that contributes to the development of the tumor. So what we kind of say is our mission is that we're really here to empower the researchers to drive that Sanger's mission of understanding biology and improving human health through these scalable genomic technologies. And I wanted to kind of talk a little bit about my career journey and how I got here. But to be honest, every time I started writing the slide, I felt very uninspired and bored. So I kept going to YouTube to watch Taeyong. So I put a picture of him instead. But if you do actually have questions about my career journey, I'm happy to hear it in the Q&A. Okay, let's get into what it takes to transform the genome. So first, a little bit of background. I'm sure many of you have heard about this, know about this in a lot of detail, but just in case, I'm going to kind of just recap what we mean by the genome. So as you can see, the people, all living organisms are made up of these clusters of cells. So cells really form the building blocks of who we are. And the components that drive cells are in the nucleus. It's our DNA. And the DNA is found in these structures called chromosomes, in which DNA is arranged in genes. You have 2 of each gene, 1 from each parent. And the genes actually encode for proteins, which again inform the structure of cells, the function of cells, and everything kind of that makes us. So zooming in a little bit. So the DNA itself is a double strand, and it consists of 4 different types of bases in an array of sequencing. So that is A, T, G, C. I'm sure many of you are kind of already aware of this. And then the DNA actually encodes for proteins by kind of arranging itself in a 3 base pair pattern, and these are known as codons. So that's there. And then the cellular machinery will translate this into amino acids, which actually form part of a larger protein. Now, kind of keeping in theme with this talk, what do I mean by the alchemist? Who is the alchemist? And I'm sure many of you know, I'm talking about CRISPR-Cas9. So for those who kind of, like Sam said, are here regularly, you probably know that we like to throw in a few questions. So the first question I wanted to ask you is whether you know when the CRISPR-Cas9 gene editing tool was first developed.
Amazing. Thank you, Nneka. I will just put that up now. So we're launching a poll. So you've got options there. You've got A, 1987, B, 2000, C, 2012, and D, 2020. So have a little guess about when you think potentially it was first developed. Do you think it was relatively recent or slightly a fair few years ago now? So I'll give you just a minute or so to put in your answers. and then we'll reveal the correct one. Okay, if anyone would like to participate that hasn't done so yet, please do put in your answer now. If not, I'm going to end the poll. So we have got, I can share the results so everybody can see. So we had 19% say 1987, B, sorry, 26% say B 2000, C 44% for 2012, and 10% for D 2020. Now there were a couple, I must admit, there were a couple of trick questions in this. So the correct answer is C 2012. So that is when the sort of gene editing tool as a way using CRISPR-Cas9 to edit a genome was kind of discovered and used in that way. 1987 was when CRISPR as an idea was first found out, and then 2020 was when the actual Nobel Prize for this discovery in 2012 was awarded. So there's a few years that are relevant, but it's 2012 is the right answer. So well done. I'll hand back over to you, Nneka.
Fantastic. Well done, those who got that right. Okay, so how does CRISPR-Cas9 work? So I'm going to get into the mechanism in the next slide or so, but what I thought maybe would help is kind of to go through this analogy. So imagine you've got yourself a Word document and you're looking for a particular word, in this case, Roango, and you want to replace it with something else. So you could use this find and replace tool to figure out where in the document you have the word, and you could automatically replace it. So CRISPR kind of works like that in principle. So rather than a paragraph, you've got your genetic code, the sequence. And then essentially what CRISPR is doing is finding the sequence that we're interested in and replacing it with the sequence that we want to replace it with. Or it could do other things like delete bits of DNA and stuff like that. So kind of as a concept, CRISPR-Cas9 is kind of acting like that find and replace function. But let's go through what it actually does in biological terms. So first, I also wanted to introduce a few key phrases that are really essential when you want to understand CRISPR-Cas9. The first is the guide RNA, and that kind of acts as the tool that searches the DNA for the sequence that you're interested in. So it will kind of bind to the relevant sequence based on those base pair codes. You've got the Cas9, which is actually a protein that complexes with the guide, and the protein itself cuts the double-stranded DNA, like in this little picture. And then you've got the target DNA, which is the sequence that you'd like to modify. So what is actually happening when we use CRISPR-Cas9 is, like I said, the guide, which is seen here, along with the Cas9 is complex. And then within a cell, the guide directs CRISPR-Cas9 to the target sequence, and the protein can now cut that region. And after you've got a double-stranded cut, there's a couple of ways the cell repairs itself. And it's critical that it repairs this because having this damaged DNA is actually going to be very detrimental to the cell. It typically uses its own repair mechanism, which leads to non-homologous end joining, which is basically when the two ends are stuck back together. But in doing so, it actually is likely to introduce errors. Sometimes you get bits of DNA deleted or random sequences are added in. But regardless of whether you get an insertion or a deletion, it alters the genetic code. And often that leads to the gene of interest losing its function. And the other mechanism is homology-directed repair. And in this case, you do the same upstream things. You've got a double-stranded break, but this time you also introduce what we call a donor template. So this is the DNA sequence with the alteration that we want to add. And then within the cell, this template is used to repair this break. And then you get finally your gene with the inserted piece of DNA. Now that's it on paper. This is more or less what it looks like in cells, especially in terms of what we do. you start off actually mixing your cells of interest with the Cas9 guide complex, which is actually called an RMP. That's that then. And then what we do is add this electrical pulse in a process called electroporation. And probably as the name suggests, the electric pulse actually leads to kind of these pores within the cell membrane, which allows things that you've mixed the cell with to actually get into the cell. So that could be pieces of DNA, or in our case, it could be the RNP complex. And then this change in the cell membrane is actually temporary. So the cell membrane can then go back to normal, but now it actually has our RNP in it. And so once they are added into the cells, the cellular mechanism can now do that process that I've just described in terms of repairing that double-stranded break, either adding this mutation, random mutation, or an accurate piece of DNA. And then there's other kind of downstream processes that I won't go into too much detail here, but I'm happy to chat to you about it after. But the final kind of thing you want to do is look at whether you've actually got the insertion of the sequence. And we do that via various methods, but often use this process called Sanger sequence, which gives you an output like this, which tells you what the genetic code is. So that's CRISPR in a nutshell. And now I kind of want to talk to you a little bit about what we do with it. And for us, especially at the Sanger, a lot of it is about understanding the unknown and really understanding biology at its basics, the fundamentals of what makes us us, what makes certain cells develop into different shapes, what gives them electrical potential, kind of all these questions is all what we can start to explore using CRISPR. And the cell line that we tend to use is called an iPS cell. So that's an induced pluripotent stem cell. It actually is derived from cells often from the skin, so fibroblasts, and then they are modified so that it reverts to a state which is a lot more similar to stem cells. And that allows it to be differentiated into an array of cell types. So you've got your neuronal cells, blood cells, bone cells. It really opens up the potential. And so we tend to edit the iPS cell and try and understand what it does in terms of developing into these different subtypes. So I wanted to give you a bit of an insight into what we are working on at the moment. So we are working with a really cool team led by Maz Hanifa, and they are interested in understanding the development of skin. And the way they've been doing it is to grow what we call organoids. So these are these 3D models which in many ways replicate certain organs. Often they're called like mini organs. You might have heard of the concept of a mini brain. So they're not quite mini organs because they're certainly not that complex, but they allow you to replicate a lot of features that you do see within these organs. So in this example of a skin organoid, you can see that initially you start off with your pluripotent stem cells. You add external factors that will stimulate growth factors and expression of different key components that are needed to drive a stem cell to become a skin cell. And through growing them under certain conditions for a certain duration of time, it starts to develop this 3D structure akin to the skin. So you can see after a few weeks, you've got these skin cells forming in this very skin-like layer. And underneath, you can see kind of what you actually get in real people, in real skin cells. And then later stages, you start to get hair follicles growing, and even you start to see hair. And so you can see here that what we've actually been able to replicate in the lab is something that takes quite a while in people. So it actually lets you start to understand the development of humans, but in a much quicker time frame. And Ferry and April in the lab have kindly allowed me to show you some of their pictures from their skin organoids. And I have to say, these are kind of the types of things that make me really excited to work here, because they've got these organoids and you could start to see, hopefully you can see my cursor, but you can actually see the hair forming. You can also see that hair, lots of little hair follicles forming. So they actually are in the lab kind of developing skin, which is, in my opinion, pretty amazing. Okay, so let's talk a little bit more about what we're doing with them. So there are a few cells that they are, a few genes, sorry, that they are really interested in learning about and what they do within these skin organoids. And so to help them visualize this in the skin organoids, what we've done is actually target the genes of interest with a fluorescent protein. So what we essentially do is we have a gene, and then we will use CRISPR-Cas9 to break it. We'll add the repair template, which has the fluorescent protein. This is just for illustrative purposes, but this is like a red fluorescence and a green fluorescence. And then if it's successfully integrated it, you should be able to shine the samples under fluorescence and see the emission of the green or the red fluorescence. And so in this case, we added a red fluorescent reporter. And you can see, you know, on the normal bright field microscope, you've got your organoid developing here. And once you see it under the fluorescent light, you could clearly see certain cells within it are fluorescent. And then they could kind of monitor these cells over time to kind of figure out what they're doing, when they're doing it, what interactions they're making, and how they actually contribute to creating these organoids. And the more you start to understand what's going on and how these are developing, the more you understand skin development in general. And in kind of looking at different key factors, different key cells, you start to develop even more accurate models that replicate the human skin to a better degree. And, you know, from this basic understanding of skin development, you could start to apply skin organoids to disease models, right? So, for example, if there's mutations of a certain disease that leads, a mutation in a certain gene that leads to an inherited skin condition, you could start to use the organoids to understand, one, how that mutation affects it, and also what you could do to intervene to hopefully try and fix things. And that eventually could lead to some cures, or more likely kind of just treatment and therapies to help alleviate those types of conditions. Another thing you could start to do, especially as you get better and stronger skin organoids, is potentially use it for things like wound healing and skin grafts. And that's kind of a process known as regenerative medicine. So this is just one example of the types of kind of projects we do. And it kind of demonstrates how we work with our active researchers. So we do the generation of the cell lines, and they could take it any way they want. So with the initial iPS cells that we generated, these guys have chosen to do skin organoids, but some people might look at neuronal development or look at the development of guts. And there are different organoids you can create to replicate these different features of humans. Another thing that we've been working on a lot recently is understanding variants of unknown significance. Now, to try and illustrate what I mean by this, I have taken inspiration from Sharon in my lab, who did a great presentation not that long ago. And I felt she did a really lovely job of illustrating what we've mean. So I've just to caveat that this has been largely inspired by her. Okay, so we've got a family, Jane Doe. She has a condition that has been found in different family members. So it appears that it runs in a family. And she now wants to know whether her daughter gets it. She does her genetic testing. It reveals a G mutation in the gene of interest. And also her mother has that gene. And, you know, the genetic testers, the scientists, can actually look at this mutation in various databases and tell her whether it's likely to be benign, which means it doesn't cause disease, or is likely to be pathogenic. It does cause disease. And in this case, the G is pathogenic, which she knows because she has the condition. Her daughter, though, has a T. And looking through the database, there isn't any information on that. So that's a variant of unknown significance. We know it's different from the usual, which would be C, but we can't say whether it's going to be disease-causing or not. So what do you tell Jane? And this is something that a lot of people face with. Whilst we do know a lot about the genome, there's so much that is unexplored. And you might have a sense of which gene is really important, but because variations in the genome happen all the time, we all have different mutations, right? And most of them are fine, they actually are benign, but some are disease causing, but you can't always tell. So we've been using a technique called saturation genome editing, and this is something the Institute is doing more and more of, and we're lucky enough to kind of be a part of it. So to try and explain this, I'm going to go back to an image I showed you earlier, just to refresh memories, I guess. So you've got your genetic code, and in these 3 base pair sequences, i.e. codons, they will be translated to the amino acids which form the protein. A mutation in the genetic code will alter the codon, and that could lead to alterations in the amino acid sequence. And that's really crucial for the structure and function of protein. So change in this could have big impact on a protein. And so in saturation genome editing, what we do is exploit that fact, but also pair it with CRISPR-Cas9, particularly using that homology-directed repair pathway. So here we've got a gene, doesn't matter which gene, and the genes have promoters, so that's where the gene expression is driven from, exons, which harbor the protein coding sequence, so i.e. all of these codons, and this intronic space, which can have regulators and other factors that help with the gene expression. And so with saturation genome editing, what we do is create a library of the HDR template. And in the library, each specific space in that genetic sequence will be mutated with each possible variant. So take here, initially we've got a G. So each library will have a different code in this sequence. So it'll have an A or a T or a C, whilst this sequence will have the G or the T or an A, so on and so on. So you create this library of all the possible single nucleotide variants. Once you've got a library, you add it to these hap1 cells. There's various methods. You could use electroporation, like I mentioned before, but there's a lot of methods. And each haploid cell will have just 1 copy of each edit. And then you just ask a simple question, will the cell survive? Has that mutation led to lots of cell death? And therefore it has detrimental effects to protein function? Maybe it has positive, and actually you get lots of cells growing who have that mutation. But essentially you kind of look at the survival rates and sequence each cell to see which edits are still left in your final pool of cells. So if we kind of go back to so if we kind of try and apply this to a more real life situation, I wanted to draw your attention to a recent paper published by Matthew Hurl's team in our institute with Lizzie Radford, and they've used this technique to look at the DDX3X gene, and that gene is known to contribute to neurodevelopmental disorders. And so using the method that I've just described, mutating every single possible base in each position of that gene, they've come across these results that once they analyzed the data, they had some mutations that led to cell death very quickly. Some led to cell death, but it took a lot longer, and you can see kind of that dropping off over time slowly, whereas the faster beaten, you get cell death in a few days. And some had no effect on the number of cells grown, so presumably these genetic changes don't actually affect the function of the gene, and some are slowly enriched, so perhaps it has a positive effect, right? And importantly, they've kind of done a bit of breaking down of what all of this means. So they found that synonymous mutations, and these are mutations in the genetic code that doesn't actually change amino acids. These were largely unchanged in that pool of cells. It didn't change the dynamics of cell growth. You've got missense, which is where you do get a change, but yeah, you do get a change. Some of these led to the cells dying off quickly, and some didn't have much of an impact. And they looked at other types of mutations. This one here is non-synonymous, so it adds a stop codon, and that has a massive effect on the protein function rate. And as you can see, these pretty much died off straight away. And importantly, they looked at whether these variations actually align with what is known about the clinical data of this gene. And there's a lot of information here, so I'm just going to point out a few. This second group of results are variants that are known to be pathogenic or likely pathogenic. And in their hands, they found that there was a lot of fast repeating variants. So these variants had clearly a very negative effect. And the more benign mutations, so the mutations that are known not to cause a disease, you found all of the variants that didn't have any change to cell growth. So if we were to apply something like this to Jane Doe, and we found that T sat in that unchanged pool of cells, what we can now tell Jane is it's likely, and remember this is based on probability, that there will be no development of disease in her case. So that's what variants of unknown significance means, and how CRISPR-Cas9 can be used to try and get a bit more information on them. Okay, so that's the stuff we do in our lab in collaboration with different researchers. So what is the world of CRISPR-Cas9 like outside of this? And first, before I get into that, I wanted to ask you guys if you have any ideas on how CRISPR-Cas9 has been used so far.
Amazing. So I will throw up a poll now for you all to vote on. So that should be launched. So how has CRISPR-Cas9 been used so far? So editing crops, editing livestock, editing immune cell for cancer therapy, editing embryos to help research different genetic diseases, all of the above. So vote on which ones. Hopefully, as I can see, I'll give you sort of 30 seconds on this one, because we'll get into most of some of this stuff in just a second. Okay. So vote if you would like to, and if you haven't done so already. But the majority of people are picking all of the above. Again, it was a little bit of a trick question from me, but yeah, essentially all of these things are where CRISPR-Cas9 is being used so far. So yeah, well done for those that instantly sort of put all of the above.
Yeah, absolutely. And I'll explore some of that with you today. So one use which you may have heard of before is using CRISPR-Cas9 to help kill off cancer cells. And this is done using CAR T therapy, chimeric antigen receptor T cell therapy. And you know, there's been a lot of kind of advertisement in recent years, because NHS are starting to roll out this more widely, and some work has been done to show that actually there has been some long-term benefits for people who have had this therapy. So it's really promising. And what it actually does is use your own cells to fight off tumor cells. So what happens is that a patient with cancer has their blood samples taken, and from that you isolate T cells, which are really important in immunology and fighting off infections. And within this, you use CRISPR-Cas9 to add the chimeric antigen receptor, and there are a wide variety of different antigen receptors which allow you to fight off different types of cancers. And also the cells are often modified further to help with the immune response. And then once you have these edited T cells that they're fully sequenced and validated, you could then re-infuse them back into the T cell patient, and then they can now bind via the antigen receptor to different cancer cells and start to lead to their cell death. And kind of similarly, but more recently, some therapy for sickle cell anemia or sickle cell disease have been developed. Sickle cell, in case you don't know too much about it, is a disease of the blood, essentially. So this image shows normal red blood cells and the sickle red blood cells. And basically what happens is there's a mutation in the hemoglobin, which is what actually binds onto the oxygen, and that actually changes the structure of the cells. And in this structure, they become quite sticky and start to clump together and start to block blood vessels. And that leads to starvation of oxygen to the different organs. And this is like really detrimental to the person and often leads to very intense periods of pain. But more recently, UK's approved some gene editing treatments for this disorder and a related disorder. And this just outlines how it works. And it's quite similar to the CAR T therapy in that you use the body's own cells. So this time you get cells from the bone marrow, which you have these forms of stem cells there. And usually you will get the sickle cell once these develop into, when you get the development into adult hemoglobin. But at the stem cell stage, what you can actually do is turn off this gene, BCL11A. And by turning it off, what you actually do is switch on the expression of fetal hemoglobin opposed to the adult kind. And the fetal one actually leads to normal red blood cells. So once you add these back to the person, as you see here, the person can then start to develop these normal red blood cells and therefore can function normally in terms of capturing oxygen and delivering it to various parts of the body. So those kind of are all really well and nice stories, but there is also kind of other elements of CRISPR-Cas9 which really start to push the boundaries and push the boundaries in terms of the science, but also kind of in terms of public perception. So I'll talk with you about some of these. First is meat in a dish. So there has been a little bit of talk about growing meat, right? And recently there have been gene edited cells which are used to kind of grow meat in a dish. So this is a company called SciFood, and what they use is muscle cells, I believe cow muscle cells. They'll edit them so they grow faster, and you can kind of scale up the production, and then they would use those meat cells, kind of blend it up and add it to a plant-based burger to kind of give it a more meaty taste. So that's quite an interesting quandary, especially for people who don't eat meat. And then kind of moving on kind of a little bit, but keeping to that theme of meat, there has been a lot of work on editing farm animals. So here you've got edited pigs, which are now immune to really, really deadly virus. And then here you've got edited cows, and you've got some edits that make them hornless, so you don't get more. It restricts the level of horn-induced injuries. Also, another group looked at using gene editing to shorten hair, which means the cows are more amenable to be reared in hotter climates. And kind of the idea behind this is really that through editing the cells, you kind of lead into less death and less waste, essentially. And just kind of a little bit of information on how you might do this in case you're interested is it's very much like IVF that you hear about. So you've got your donor female, in which you get your eggs, you fertilize it. The fertilized egg can then be edited with CRISPR-Cas9. You grow up the embryo somewhat, and then you get a little bit of that to sequence, make sure you've got the edit that you're interested in, which is just hair. And then this embryo can now be added to your surrogate cow. And then the cow that is born from that embryo is your edited cell. Sorry, I think my computer crashed a bit. Let me pull up my presentation again.
Yeah, we've just lost your slides there. Maybe whilst we're waiting, we actually have a poll that we were going to ask just after this. Yeah. So let me throw up that poll, which is just quickly to ask you where sort of you might potentially feel around genetically edited sort of food. So this poll is asking basically in the US as of 2023, there have been 2 approved basically foods for consumption. So one's a pig that's been genetically edited, and one is a salmon. Would how likely do you feel currently about sort of your consumption of genetically edited food? We'll give sort of 20 seconds on this one, just because I'm conscious of time and so much to get through still. So yeah, just a quick sort of one. If you don't feel like you want to participate in the poll, this is totally fine as well. But we've got a real spread, to be totally honest, in the room. So the sort of majority is kind of saying, probably, sorry, but we've got a real split between all of the answers. So it kind of just highlights that it's quite a complex issue, and also one that's quite hard to distill, sort of about public perceptions on this type of stuff as well. So yeah, thank you. I'm going to end the poll and hand back over to you now, Nneka.
Cool. So moving on from kind of looking at the use in meat production, there's actually been a bit of research on using edited animals for transplants. And so recently, a gene-edited pig was created, and the kidney was actually transplanted into monkeys, and they did see long-term survival of these monkeys. So very quickly, the process they used is somatic cell nuclear transfer. So if any of you remember Dolly the sheep, this was the type of method they used. So here they'll get a cell from a pig, often from like an air punch or something. They'll make a modification, and then they'll pop it into a oocyte, which is essentially a matured egg cell, where they've taken out the original nucleus, they transfer the somatic cells, and then that could develop into the embryo, which is then implanted in the surrogate mother. And then the piece of work itself, I will try and kind of explain this fairly quickly. They created several pigs in this manner, and they either knocked out some key cells involved in immunology, or they also kind of added other factors that were important for the survival of the kidney. And then they looked at, when they transplanted it into monkeys, what type of survival they got, and kind of increasing the different mutations. I think they had 69 mutations in total, so it's this one here. They saw survival. So you've got your survival and the times after transplant, some of them survive up to 2 years. So showing kind of that the cross-animal transplantation could be effective. And kind of more closer to home, let's say, recently there was a procedure which used gene-edited pig liver. So it was edited again on key immune functions, specifically for the human immunology, sorry. And they actually didn't transplant it into human, but they actually did a kind of they used it as a filtering system, which was attached externally to somebody who had lost brain activity, and found that it did actually work. It managed to filter the blood, but just temporarily. And this is the lovely pig that donated its organ, let's say. So moving forward, with all of that, a question that often arises is, can we make gene-edited babies? Short answer is yes, and it has already happened. And this was massively controversial because there's actually a lot of risks associated with this. So with that, I'll ask you, what type of risks you think might we be facing here?
Yeah, so we just launched a poll where you can sort of type in any thoughts that you might have with sort of risks of sort of using gene editing tools. Nneka, I think you might have mentioned a few of them before. So yeah, just type in a response if you'd like to. Again, we'll give sort of 30 seconds or so on this one, just so that we get through the other stuff. Okay, and I saw a couple of questions, which was around about what are some of the challenges with this stuff. So this kind of goes into
Okay.
some of that. So yeah,
Yeah, I
hopefully.
think that really nicely links.
So yeah, thank you. I can see already sort of 20 or so people have put in some answers. I'm going to end the poll, but have a think if you are still sort of typing an answer. I can read some of the sort of the answers. So it could off-target effects. Lots of mentions around sort of like cultural and ethical issues or unexpected results due to the mutations. Yeah, lots of stuff to do with unexpected mutations.
Yeah.
Also a lot about the unethicalness of it all.
Yeah, so that's exactly right. And actually in this article, they mentioned it being messy. And I think that's kind of really the big part of it. So earlier I gave you this kind of analogy for gene editing, but it's actually not that simple. Nothing ever is, right? So it's actually more like this. So you might have a sequence that you're interested in, but that sequence might arise in other parts of the genome. And you run the risk of modifying those parts as well. And if it's in a critical gene, you don't know what type of other disorders and diseases might occur as a response. So with the technology isn't quite there yet that you could be really confident that he's just made the edit that he hopes for, and we don't know the long term what other challenges these kids might face. Another thing to consider is this gene in specific, once it was mutating, it offers some resistance to HIV. It also, that mutation leads to predisposition to other illnesses as well. So actually in preventing one disease, you actually might encourage another one. So yeah, there's just a lot of kind of unknowns here. And as a result, actually, he was sentenced to prison here. Which makes sense. And the whole community was really, really kind of up in arms, trying to kind of start to think about the ethics of this a lot more seriously. So just to kind of to round off, I wanted to touch upon AI, because obviously AI is just so important these days, and like every other day it seems like you're getting information on just how much AI could do. And actually in this field, it's also very important and has a lot of potential benefits. And, you know, this isn't my area of expertise, but I think it's still important to highlight. So one thing is actually using AI and machine learning could actually lead to gene editing being a lot more accurate. And you see a lot of papers that are focused on machine learning being able to predict the outcome of CRISPR-Cas9 editing, being able to reduce or predict off-target efficiencies. And kind of the more and more we do these experiments, the more they could be added to these databases and we get more precision, right? And so perhaps we get to a point where actually the edit of the babies wasn't so dangerous because you could be a bit more certain of what the edit was. But obviously that doesn't change all of the ethical concerns with that. So I'm hoping that we're still way off that. But, you know, AI is actually making things a lot more possible. The other thing that I really wanted to talk about is this really cool piece of research by Deborah Marks' group, I think in Harvard, I want to say. She actually did a talk here recently and it was great. But basically what they've been able to use is machine learning to start to predict variants. So they have developed this tool which allows us to predict whether a variant, and actually they focused on SARS-CoV, whether they were able to escape the immune system and actually become quite a prevalent variant. And they based this on various things. So one is fitness, which just means will the variant lead to the viral death? Will the variant be accessible to antibodies? So will it evade the immune system? Also, does it affect the antibody binding site? So how effective will antibodies be in disrupting it? And pulling all of this information together, they were able to create a really strong model, which when they actually inputted the data that we had related to SARS-CoV into this model, they were actually able to predict quite a lot of the variants that we actually did see. And actually, it was more efficient than any other models or any other kind of lab-based work. So if it was implemented earlier, that would have just given a lot of time for us in the lab to actually start to understand, well, what do we need to combat it? How do we develop vaccines? What vaccinations will start to become redundant and stuff like that. So it can actually be a very, very important tool for predicting kind of epidemics and stuff like that. But also kind of going back to the variants of unknown significance, it can actually also be used to predict whether a variant will be pathogenic, cause a disease, or whether it be benign and not cause a disease. So there's a lot that can be that we can get from AI. Okay, and just the final slide I wanted to touch upon is equity in gene editing. And I think this is a really important thing to consider. And I'll show you why. So we've got sickle cell disease, right? And I've talked about the really exciting piece of research and trials that have been undertaken, which suggests you could use gene editing-based therapy for it. And I think we could all agree this would be really fantastic, right? But then you have to kind of think a little bit deeper. Medical injustices, right? So sickle cell is often seen in Black communities, which, you know, typically have encountered a lot of medical injustices, right? Whether it has been kind of being included in clinical trials without their knowledge, whether it's discrimination felt in the medical system, there has been a lot of issues here. And so I think creating another therapy is all well and good, but then will it negate the injustices and inequity we see in medicine, or will it exacerbate it? The other thing is cost, right? This therapy, I think I saw was around, I think 2 million. Now that will definitely go down, but regardless, it's going to be very expensive. And who could actually afford that, right? Surely it's going to be people who have money, and that wealth gap would then start to increase. And in turn, that would lead to more disparities, I think, in the medical system. Specialized centers. So this isn't a simple fix. It's not just a pill, right? You need really specialized centers, and actually there's not many of those around. So I think I saw something saying there's about 200 in the US and like 3 in all of Africa. And Africa has a lot more sickle cell cases. So who's actually going to be able to be treated? And finally, it's environmental thoughts, because whilst sickle cell is well characterized, sickle cell has also been linked to resistance of malaria, because actually those cells in that sickle format doesn't really allow parasite growth in the same way. So you might get to a case where you've eradicated sickle cell, but now another disease kind of becomes more prevalent. So a lot of things need to be done to kind of think about the relationship between the two. And then finally, I put this image of Angela Saini's book, The Return of Race Science. And I always think when you have things like this, you know, a disease associated with a particular demographic, it can be a little bit hairy, because it implies that there is a biological difference between the races. There's a genetic difference. But actually, the bigger thing is the conditions that allowed this disease to develop in this case, the malaria, and the fact that sickle cell actually was a benefit to people. So I'm always kind of aware of what type of conversations can arise. And you know, COVID was a great example, right? Where you saw, you know, people talk about certain races being more predisposed, when actually the conversation should have been about social and economics and conditions and stuff like that. So that's just something to consider when you think about gene editing. So with that, I'll say thank you. And yes, I'll just leave you with this quote from one of the pioneers of gene editing, who also acknowledges there's a big risk that there will be a lot of inequity associated with it.
Amazing. Thank you, Nneka. That's yeah, thank you for going through all of that and ending on that really interesting, thought-provoking message. I guess on that same regard, just quickly before we get into into the Q&A, is that on that equity front is really important to know about the voices of the people that are most affected by this type of stuff. So on our site, we ran a citizens jury on genome editing with 21 jurors that all had lived experiences of genetic conditions. And that link I've just put in the chat, you can go to that page, you can watch a 20-minute video that sort of explores some of the emotions and feelings and opinions of people that have real lived experience of certain genetic conditions, and their ideas about whether or not genome editing should be involved in some way in sort of helping them and to with certain conditions and the risks and the tension that you can see with everybody in that room. So I would definitely recommend checking that out. We've got literally only a couple of minutes to do Q&A. So if we don't get around to any of the questions that you've put in the chat, don't worry, we will get responses to you in some way or another. I'll pick out a couple quickly for you, Nneka. So one is saying, how is CRISPR delivered to the patient? Is it transfused in the patient's blood intravenously? Is it given in an inactive form?
Yeah, there's a few ways. So kind of in that kind of CAR T therapy, you do get the infusion. There are other ways, for example, viral transduction. So when it's packaged up into a non-harmful virus, that's another method that people have been exploring.
I think this is thank you. Sorry, I'm rushing through these, but yeah, thank you. When this is a really interesting one and thought-provoking, I can imagine for all the group, but when do you think gene editing will be a reality routine clinical practice? How far off do you think we are from it becoming routine?
You know, I really can't answer that question. I mean, it is already a reality, right? But I think the only thing that it's dependent on is getting costs down. But I have to say, I think that happens fairly quickly. So I think for kind of the really kind of serious illnesses where there's a very clear biological understanding, I think that would be fairly soon. I mean, CAR T therapy is getting more and more used in the UK alone.
And this kind of links on a little bit, but it's sort of like, I guess, where does this technology end, if that makes sense? So there's a question where it's like, so why can't we cure any mutational disease with CRISPR? And I guess it might come back to some of those risks that you were talking about, that we talked about earlier.
I mean, I think the risks are the major thing, but I will say that, you know, it is actually currently being used. So I think it's just more about expanding what you could use CRISPR to treat rather than whether it's going to happen.
And someone's just put, is there any way to predict any of the side effects that happen with gene editing?
So there's certainly plenty of ways to predict the off-target effects. And so with that, if you know kind of particular genes or mutations cause certain disorders, you could probably get a nice prediction of that. But to be honest, if you are kind of looking at whether you're likely to get these off-targets and you do see something that's serious, you wouldn't go forward with it, right? You'll choose another guide. So you probably won't be able to predict anything that's unintended that you can't really explain, but you can get a lot of information about off targets.
Okay, thank you. I'm noticing the time, so I'm sorry for any of the questions that we don't have time to get to. I will make sure that I'll send an email round and I'll be able to add some places where you can get to find more information about all of this stuff. But before we end, I also just want to highlight the next session that we have, and you touched upon a few of the points that we're talking about. So we've actually got some people from the Hanifa lab coming to talk about their use of creating basically a human cell atlas and understanding cells. And we're going to use the example of skin cells later on. One. Yeah, so if you want to sign up to that, here is the link to do so. Please do, and I look forward to hopefully seeing some of you there. Yeah, so I just want to round off and say thank you so much to Nneka for spending your afternoon with us. As I say, any questions that we didn't get to, I will make sure we get you answers. But yeah, thank you everybody for joining us this afternoon.
Bye.